Hello, this is DrPaulCottrell, and I'm going to be talking about part 3 of the pseudouridine. This video is going to be about the metabolism of pseudouridine and the methylated version of pseudouridine, which is tied to Weissman and Karako's contribution to pseudouridine and the messenger RNA platform. So what I'm going to do is I'm going to do another whiteboard here. As you know, I did 2 other videos. The first video was going over just the basics of pseudouridine and uridine and how they improve the structure of tRNA, mRNA, rRNA, and snRNA, and then in some cases non-coded RNA. And then on the second video, I went into more detail about the actual enzymes that turn uridine into pseudouridine. So pseudouridine, again, looks like this. Okay. I gotta draw right. Right, and then you have hydroxyl groups. And then you have the 6 ring. This is the pseudouridine. Symbolized as this. And this is created by the pseudouridine synthase proteins. All right, pseudouridine synthase proteins, and there are many different ones. 1, 2, 7, 8, and they all find a region within the messenger RNA or the tRNA or whatever RNA you're dealing with. It finds a location and then takes the uridine and converts it into pseudouridine by moving this piece or rotating it around this axis and flipping it, and then you end up getting, I missed a double bond there, and missing and getting the pseudouridine. So, and that enzyme is this pseudouridine synthase. Okay, so with that said, we are going to now, and this is all post-translation, meaning post-transcription, meaning you're going to have an RNA that's something like this, right? And after applying the PUS, you're going to get some that, and certain ones will recognize certain topologies, certain sequences for the RNA. It may convert this into this. Let's say, for example, PUS1. Okay, but if let's say we were going to use PS5, for example, you may end up getting something like this. So different PUS will change different uridines into pseudouridines. Now, with that said, and this is all post-transcription, and then from there, then a protein can be made. Now, what I want to go over is the metabolism of pseudouridine. All right. Again, you have the ribose hydroxyl groups. And now you have that 6-ring flipped uridine or uracil. This is this, right? So this is going to be the pseudouridine. Okay. All right, now we have something called a pseudouridine kinase. Now what that does is it adds a phosphate group to it. So we have something like this, right? Okay. And we have that ring. Okay. What will happen is that the pseudouridine kinase is going to modify this, okay? And it will turn it into, remove the hydroxyl group, and it will put a phosphate, okay? So there's going to be, let me get some space here. So pseudouridine, I'll just kinase. So this is the P, this is an O, negative. Okay, so we've added a phosphate group to the ribose. All right, to this ring here. Okay. All right. Now we have we can have a reverse reaction. Okay. So we have pseudouridine post-transcription changed by a pseudouridine synthase. That's how we get the pseudouridine. And then the metabolism breaking down the pseudouridine as it's recycling the RNA is done by this pseudouridine kinase. It adds a phosphate group to it, and then we have this reverse enzyme. So PSIMP monophosphate, right? And it's glycosidase. Now what that does is that it takes this enzyme, breaks apart this bond. Okay, so I'll draw it. So what happens is that you end up getting a ribose so it helped this enzyme, this glycosidase, breaks the bond by adding a hydroxyl group, All right. And you still have your phosphate. Okay. This is what is called ribose 5P. Okay. And then you also get uracil, which is the top group. The nitrogen and the hydrogen, the nitrogen and the hydrogen. And then you have this double bond here. Okay, and this is yours. Okay, so that's how we metabolize pseudouridine into uracil and ribose 5-phosphate. Okay, now what I want to do is go and talk about briefly the methylated version of this, which is the contribution of Weissman and Caraco. Okay, so what they found is that if you could put if you could have messenger RNA and you have a sequence, right? That's foreign. Let's say this is a foreign sequence, messenger RNA sequence. 5', 3'. Okay? And let's say this is foreign. If this gets into the cell, then the cell is going to say, oh, foreign, attack it. Okay? Oh, foreign, attack it. Well, what they found out, and this is through the innate immune system, what they found out is that if you make methylated pseudouridine, and you change this, M means methylated pseudouridine, you change this, and not all the uridines would be methylated, then you would fly underneath the radar of the innate immune system. Okay? Now, there are two ways to remember what I said when we take uridine. And we convert it to pseudouridine. This is all post transcription, meaning messenger RNA with uridine, and then messenger RNA converted through the PUS, right? Then with some pseudouridine, right? Some pseudouridine. Okay. Because of the topology, depending on what type of enzyme you're using, that's the PUS. Okay. And that was all described in the last video. Well, there are two ways to do what Caraco is doing. Okay. And Weissman. Okay, there's two ways. Right? Okay. The first one. Post-translation transcription, I should say. Post-transcription uridine to pseudouridine for RNA. And it doesn't matter what kind of RNA. It could be the message. In this case, they're dealing with messenger RNA. But this would actually work for transfer RNA also. Okay. Post-translation. Then you take, so there's two ways for methyl pseudouridine. Okay. So the first way is to do the post-translation, post-transcription of the RNA. And that basically means uridine to pseudouridine using PUS, some POS, some pseudouridine synthase. Then the next step would be to convert the pseudouridine to methylated pseudouridine. Okay. Now, what is methylated pseudouridine? Methylated pseudouridine is the ribose, right? With the 6 ring and the difference, instead of having a nitrogen, hydrogen, and nitrogen, hydrogen here, the difference is that this is changed to this has a CH3. Okay. And so this here, to convert pseudouridine to methylated pseudouridine, which is this, you need something called a methyl transferase. Okay. And so you take that pseudouridine, use the methyl transferase, and then make off of that code. So you had code that say was this, right? And you went through and used the PUS. Now you have pseudo. Okay. Let me turn this into a pseudo. Now we're going to use methyl transferase. And now we've converted and made that code AM pseudo. So methyl pseudo, methylated pseudo, CC methyl pseudo. Okay. That's one way to do it. Another way to do it is as the codes, as you're synthetically making this code in vitro, let's say the code is this. Okay, that's the code without doing pseudouridine or methylated pseudouridine. All right, and you have certain concentrations of A, certain concentrations of U, certain concentrations of G, certain concentrations of C in your system, in your solution. Okay. Now, the idea here is for number 2, it's not post transcription, meaning you don't have this sequence first. You're gonna end up with this sequence. Okay. Now, the idea here is that to make the DNA, or you have DNA, right? To make the RNA, you have to go through some sort of RNA polymerase, right? RNA polymerase. And that will make the RNA. Okay? Now, if you have an RNA polymerase that can recognize the methylated pseudouridine, then you can insert the methylated pseudouridine without the post-transcription. Okay, so the key is the pseudouridine to be able to be used with the polymerase. Okay, now if that can be done, what will happen is that you will now build up a sequence that will start. It'll start with A, right? It'll start with A. It'll have the methyl group G, C, C, and the methyl. Okay. Now, what you can do is that you can kind of randomize this. So not all the uridines are going to be replaced by methylated pseudouridine. What you can do is you can play with the concentrations. So your methyl pseudouridine concentrations can be much, much higher than your concentrations of uridine, and therefore the percentage that are incorporated, the percentage of methylated pseudouridine will be much higher than the percentage of uridine in the RNA. Okay, that's one way to do it. Okay. There's also a third way that I'll go over. Now, and so by playing with these concentrations, if you go 50-50, if you had a 50-50% concentration of pseudouridine methylated and let's say just uridine, then what will happen, you have 50% also. And you have a 50-50 chance of this being incorporated, right, as a pseudouridine, 50% chance, and methylated, and this being a 50% chance. So most likely in this case, we would have something like a methylated pseudouridine, a G, C, C, and then maybe a uridine, because it's 50% chance. So you can kind of randomize it. All right. Now there is another way that can happen. All right. Now the key here is that there's a polymerase that is able to incorporate the pseudouridine to put it in by changing these concentrations that are in the flask. Another way to do it, third way, is to change the concentrations of the actual methyltransferase. And the concentration is the POS. Okay, remember POS is the pseudouridine synthase. So in the cell, naturally, normally we have uridine, and then it can convert to pseudouridine with the POS. Okay. So if you wanted to make the messenger RNA to have methylated pseudouridine, if the goal is to have methylated pseudouridine, another way to do this, is to change the concentrations of the POS and the methyltransferase. So what that basically means is that you're building up, as your messenger RNA is building up, you have a certain number of you have a messenger RNA that now has a sequence of this. Okay. Then the next step is that you have a high concentration of POS if you want all of your uridines to turn into pseudouridines. So you'd have to have a high concentration. But if you want only a small percentage of them, then you'd have a low concentration of POS. If you did the low concentration, then what would happen is that you would get something like this. Then you would have the methyltransferase coming in. All right. And that methyltransferase would turn the pseudouridine into methyl pseudouridine. So if we had this concentration of methyl transferase, you would have this change, and it would not change this. So let me go back so people can understand. You can play, so there are a couple different ways to do this. Okay, we're talking about methyltransferase now. Okay, so there are really three ways. Okay, the first way is more similar to what happens naturally in the body or in the cell, post-transcription. So that means is that you have normal RNA without pseudo or methylated pseudo. And what happens is that now you have to convert the uridine into pseudouridine, and then from the pseudouridine to the methylated pseudouridine. Okay, and to convert the uridine to the pseudouridine, you're using PUS, and from pseudouridine to methylated pseudouridine, you're using the methyltransferase. The second way to do it is not a post-transcriptional method, which basically means is that you are playing around with the concentrations of your methyl pseudouridine that has been pre-made, all right? And you're adding it to this mixture that's going to be growing this, or your system that's going to be growing this messenger RNA. This would work well with the in vitro, not in vivo. Okay. So you add in a polymerase that can use these methyltransferases. These polymerase that can use this methylpseudouridine. Okay, so now what you're going to have here is that you can play around with randomly where these things are being placed based on concentration levels of either your methylpseudouridine, higher relative to the uridine, and therefore you're increasing the chances of methylpseudouridine in your RNA sequence. Now you got to remember the difference between the post-transcriptional method in vitro versus the first method that I showed you is that this method uses the POS, right? A certain type of enzyme that recognizes certain type of pathology or certain type of sequence and inserts. So this method is a little bit more surgical, a little bit more exacting. This method is random. There's a randomness relative to the concentrations. Now in this method here, this method is like the post-transcription method, but you're playing around with the concentrations of the actual enzyme. And by doing that, you are increasing your how much is being incorporated. All right, you're kind of fine-tuning it. Now you got to remember that the POS is more specific. There's some generality to it. Some POSs have a general, specificity, and some of them are much more specific. All right, so some locate generally, some locate very specific, and that's been covered in the previous video. So this method is more changing kind of the concentrations here and being able to use some randomness to allow for it to happen. Now, what is interesting here is that the more concentration of, let's say, methyl transferase that you have, the amount of methylpseudouridine in the RNA will increase. Okay. Okay, now why do they do this? Why they do this is for two main reasons. Okay. One is that the pseudouridine, the methylated pseudouridine, can inhibit the innate immune system. The term they use by Weissman and Carco is non-immunogenic. Non-immunogenic. Okay. Now, I covered this years ago. I don't know, 2 years ago, 3 years ago, something like that, and why this is. Okay. Now, what is happening is that in the cell, I'm just going to briefly mention it. In the cell, especially with macrophages, but in the cell, the cells get infected, let's say, by a virus or a bacteria, right? Or a bacterium, right? and they have features on it, and they can activate what is called a toll-like receptors that are on the cell. Toll-like receptors. Okay. Now, when certain types of toll-like receptors are activated, it will determine if it's a bacteria, if it's a fungus, if it's a virus, right? So it'll depending on which toll-like receptor is activated. Now, a lot of times it's because of either the proteins, the glycolysis, that the glycoproteins that are on the actual pathogen that are activating the toll-like receptor. But the cell, when it gets some of the DNA or RNA of a foreign product, like a bacterium, it can detect that it's foreign and move and tell the cell to go into a antimicrobial state. Okay, and this is where this interferon alpha and beta come in. And when interferon alpha and beta starts to get secreted out of these cells, the surrounding cells will go into a pre-apoptotic state. They get activated. They get like pre-activated. So if something does happen, they go in and kill themselves quickly. All right. Well, what Weissman and Carrico found was that when they injected RNA into the cell, many times the cell would pick it up and start to break it down and and view it as foreign and go into and do a destructive mode. This cell will go into apoptosis, die off. It'll try to kill itself. It'll try to kill itself, but it'll also go into a shutdown of this So to make these RNAs, or to read the RNA and turn it into protein, it needs something called elongation factor 2. Elongation factor 2. Okay. So when the cell detects a foreign product, it will phosphorylate the elongation factor 2. It'll phosphorylate the elongation factor 2 and prevent. And what it does is it prevents protein synthesis. Proteins cannot be created. So that's this is their antimicrobial state. Well, what Carrico and Weissman found out was that if you have pseudouridine, methylated pseudouridine in the RNA, and you put it into the cell, it does not phosphorylate the elongation factor 2 and activate pKa. Okay. So what that means is that when you have the messenger, or when you have a messenger RNA that has methylated pseudouridine in it, maybe not everywhere, but in it, one, it stabilizes the molecule, but it also prevents phosphorylation of elongation factor 2, which means that proteins will be created of messenger RNA. That's their big contribution. Okay. All right. So I'm going to create another slide. So the messenger RNA that Weissman and Carrico needed had to have this methylated pseudouridine. And that methylated pseudouridine does a couple things. One, stable. It stabilizes stabilizes the messenger RNA structure. I covered that in the first video. Two, it prevents the cell to go into an anti-microbe state by preventing it. It prevents the the phosphorylation of the elongation factor, which leads to RNA making foreign or making protein. Okay, so they're flying underneath the radar of the innate immune system. Now, another feature of the pseudouridine is that, remember what I said, you have the messenger RNA, and in some areas you might have this methylated pseudouridine. Not everywhere, but some places. Well, you have the ribosome reading and making this protein, right? This is the protein. This is the large ribosome, this is the small ribosome. And what they found out was that by having, not everywhere, but having the methylated uridine in certain areas spaced out, you can slow down the read of the messenger RNA, and that helps with the protein folding. Because if it's read too fast, this protein may, if it, like for example, I'll try to draw. Right, so you have this ribosome, you have the messenger RNA, and it's creating a protein. If this is reading quickly, okay, let's say the velocity is elevated. All right, this protein may turn into something like that. Okay, but if you slow it down, if you slow down the velocity just a little bit because of these little speed bumps, right, then what will happen is the protein may actually fold more correctly. In addition, not just helping with the folding by changing the speed. So the methylated pseudouridine can slow down. It slows just a little bit, slows down the ribosome. The ribosomes, the large and the small. All right, and that will help with creating protein, whatever is programmed inside that RNA. Helps create protein. Okay. In addition, what they have found is that this is inconclusive here, but some data is suggesting that the methylated pseudouridine is preventing. We get sometimes mistakes, all right, in the read, and what type of amino acid is incorporated into. These are built up as amino acids, different amino acids, okay? Well, as the transfer RNA comes in, and there's these three different cavities, and it transfers out, and it's building up this protein. Sometimes the read, certain nucleotides can make a wobble, okay? And a wobbling basically means that there is a little bit of play within the structure, within the binding of the ribosomes, and the messenger RNA and the tRNA all together. Because there's a little bit of tolerance there, that something can be mistakenly put in instead of the correct amino acid. Okay, so let's say, for example, maybe glycine, instead of glycine, it's isoleucine. Okay, something like that, right? Maybe not exactly, but you get the point. So some of these wobbles can be happening. Now, there is some thought that that there is less wobbling, which would reduce protein incorrect amino acid, reduce incorrect amino acid in the protein, all right? In protein. But that still needs to be further investigated. So those are the three basic reasons why they use the methylated pseudouridine. Now let's go back to what I said here. Okay, so this is pseudouridine without it being methylated, and we have different POSs, pseudouridine synthases, that are specific or sometimes general, but can change a uridine into pseudouridine. And this is post-translation. So you would have, a normal, you have the uridines, you use the POS, it recognizes a certain sequence, and then it'll switch it to a pseudouridine. All right, and then you may use another type of POS, and it may recognize a different location to switch from a uridine to pseudouridine. Okay, now we can use, once we have a pseudouridine, we can metabolize it, right, by adding a phosphate group to it, and then that phosphate group can end up, well, the phosphate group can be used to incorporate it into the RNA, but the whole point here is that how does it metabolize? So you have a pseudouridine, right, and we can add a phosphate group to it, and then we can have, which is not reversible in this case. This is a one-way thing here, and then there's a reversible reaction using a glycosidase, the PSIMP glycosidase, and that basically breaks apart your ribose 5P and creates a uracil. And then these are recycled. These are recycled and can be reincorporated into making more RNA. Okay, now here I am showing the post-translation. So you have this post-transcription. So here you have the base, you have a base sequence, starting sequence, and then using the post-transcription of POS, you're now modifying those uridines, this uridine to this, depending on what type of POS you're using. Not POS, PUS. I don't know why I'm saying POS. So PUS. So P meaning pseudouridine synthase. Okay, next. Now, there are 3 ways for the methylpseudouridine to happen. The first way is a post-transcriptional way, which is similar to what's going on in the body. And that is, okay, first you have a base, right? You have code, RNA code that has a uridine in it. And then you use the PUS to make the pseudouridine based on some sort of sequence. And then you can convert those pseudouridine to methylpseudouridine based on using methyltransferase. Okay, so here you have a base sequence, you use POS, it changes the uridine to pseudouridine, and then you use the methyltransferase to make the methyl. And by changing this concentration, changes the and the length, changing the concentration and the length of the reaction, the time of the reaction, you can change the ability of how many of the uridines how many of the pseudouridines turn into the methyl pseudouridine. Okay, now for the non-transcriptional way to do this, or the non-post-transcriptional way, the idea is, the thought is, well, you can have different concentrations of the nucleotides, all right? And you can do this in vitro and have a RNA polymerase. So it's reading a DNA, it's creating RNA, right? So this is a polymerase today, it's creating RNA, and it can incorporate methylated pseudouridine. Okay. Now, by changing the concentrations, you can change how much percentage, relatively randomly though, how much of the methyl pseudouridine will be incorporated into the RNA when you're looking at higher concentrations of methyl pseudouridine to, let's say, uridine. Okay. So if you have really high concentration or a higher percentage, then you'll have a higher percentage of the methylpseudouridine being incorporated in the RNA sequence, because this RNA polymerase can incorporate the methylpseudouridine. If you don't have a polymerase that can do that, then you can do this method. Okay, this is post-transcription method. All right, and this is playing around with the concentrations of the PUS and the methyl transferase. Remember, the methyl transferase turns a pseudouridine into methylpseudouridine. Now, the PUS can be general or specific, right? And we went over that in video 2. And the messenger RNA will have this base sequence with the uridine in it. Then with extremely high concentrations of PUS, you would have extremely high concentrations of the pseudouridine, assuming that this is general. If you have extreme high concentrations of, let's say, specific, then you're going to have very specific locations that are going to have a pseudouridine. Okay, so you can play with these concentrations to change the speed and the length. It's not just the concentration of the enzyme, but it's also the time of the reaction. You can increase or decrease the time, and that will change in how much is going to be incorporated into the sequence. Okay, now the next feature is here. Okay, okay. There are two main reasons, with a minor third reason, why the methylpseudouridine is used. The biggest reason is due to the fact that methylpseudouridine inhibits the innate immune system by circumventing the recognition from the toll-like receptors and preventing the interferon alpha and beta pathway, which prevents the killing of the apoptosis of that infected cell and the signaling to surrounding cells, adjacent cells, to go into a pre-apoptotic phase. This also prevents the phosphorylation of elongation factor 2. Okay? When you prevent that, then what it does is it's allowing for that foreign RNA to be able to turn into protein. Now, another reason is that it stabilizes the messenger RNA. We showed that in video 1, where you can have pseudouridine stabilizes the structure of the RNA much better than just uridine. So if you have stability here, if you have, let's say, concentration of, and this is stability, right? Or, it's actually a Hertz structure, even the Hertz level. But, the actual Hertz level, higher Hertz level, stronger structure. So more concentration of pseudouridine in important areas of the RNA, depending, like, for example, transfer RNA or messenger RNA, then the Hertz level is going to go up. Okay, the structure is going to be better. It won't break down as easy. Okay, in general terms. Now there are some probably situations where it may make it worse if you have too much. It may actually be something like this. But in general terms, most of the time, it's going to be more structurally sound. Now, when you methylate that, you're going to also have that same structural integrity. So it stabilizes the messenger RNA, makes it a little bit more robust. And it prevents, like I said before, it prevents the cell from going into the antimicrobial phase. Now, the weaker third reason is the slowing down of the protein. All right. You have these like kind of speed bumps, these methylated pseudouridines, and it slows that machinery, the ribosomal machinery, the transfer RNA machinery, and as it's building up that protein, it's giving more time for it to fold. Okay, so if it reads too fast, you may get something like this. If it reads a little bit better, you get what you're looking for. It reads a little bit slower. So the velocity of the read, so increase in incorporation in the RNA and RNA. All right, RNA. The velocity of the velocity of the read off the ribosome will go down. And when you have the methylated pseudouridine in the RNA, an increase of the number of nucleotides that are like that, then what will happen is that that will increase. Let's say the y-axis is going to be proper protein folding will increase. Okay, but again, probably in reality, it's more like it gets up to a certain point and then it has a negative effect at certain levels. So there's a sweet spot. This is a reason why you play around with the concentrations. All right, now the one that is kind of related to the slowing down of this protein folding is the wobbling effect. All right. Does it wobble a little bit more to slow it down, or does it hold on to the RNA tighter and it's just going over that speed bump to allow for the protein folding? This is in question. It doesn't seem that the messenger RNA with the methylated pseudouridine does not have an increase in incorrect amino acid incorporation. Does not. Okay, so it does seem as though that there seems to be less wobbling but that needs to have further investigation. There needs to be a little bit more understanding of that. But there does seem to be this capability of the methylated pseudouridine to slow down the process of the read to help with the protein to be created. Okay. All right, so now I think in the next video will be the video that's dealing with the cancer specifically to pseudouridine upregulation and stabilizing the RNA to allow for better protein synthesis in, or more stabilized protein synthesis in tumors. Okay, now what I'm going to do, so there will be a total of 4 videos for this whole series. So I'm going to stop my share here, and then okay. So you can go to my store, the-studio-reykjavik.com. That's the-studio-reykjavik.com, and purchase the health products that I offer, the supplements I offer. But you can also donate on my homepage at the very bottom. You can donate through Stripe or PayPal. That will help to cover my news coverage. And you can get all the descriptional links under every video that I do, to get to the website or to the different channels that I have. But what I offer on the store is nano silver. This is the structural nano silver. I have it in the toothpaste form. I have it in liquids, which is the Health Max 35, the Health Max 14. I have it also in gel form. I have the soaps, different scents, lozenges, the structural nano silver and the lozenges. And I have also 2 other types of liquids, which is the alkaline version and then also an extra strength gel. But I also have a probiotic from Amio. It's in a capsule form, so very useful. Helps with the gut biome. I have a multivitamin, a great multivitamin, and these applicators. So it's like a dropper form, a nasal spray, and a regular spritzer. So, please, it's important to neutralize pathogens, and you need to do that on a daily basis, honestly. Great way to do it is use the toothpaste. That's going to neutralize the pathogens. It's going to reduce the plaque, because it's plaque that makes the pathogens, and reduce that gum inflammation. And when you have proper oral hygiene, you're going to reduce your cardiovascular problem. Cardiovascular disease, for many people, is correlated to oral hygiene. Now, I also take Health Max 35. This is a liquid, and what I do is I take a teaspoon of it a day. I swish it in my mouth. I gargle with it about 60 seconds each way. So 60 seconds for swishing, or 30 seconds swishing, another 30 seconds with gargling, or 60 seconds. It doesn't really matter. And then you can swallow it. And then what's happening is you're neutralizing the pathogens that are in your mouth, in the back of your throat, and down your esophagus. Then what I do is that I use the silver gel. I put it on my hands, around my mouth and around my nostrils and inside my ears, lightly coated. And you can use this also as a skincare product at night, where you just put it on your skin, on your face, and just leave it there until morning, then wash it off. The soaps, they're of different scents. There's the charcoal tea version, the peppermint, the lavender, the oatmeal. Lemongrass happened to sell out, so I'm going to have that in stock soon here. And I have this sweet menthol right now for lozenges. So check out those products. Those are really important to neutralize the pathogens, and that's the first pillar of the protocol: neutralize pathogens. The second pillar of the protocol is a very strong antioxidant. C60 is an extremely powerful antioxidant, and what that does is it's going to improve your mitochondrial health. It's going to improve your mitochondrial health because it's reducing the stress that's going on in the cell. By reducing the stress that's going on in the cell, you are preventing your cells going into necrosis. All right? And by doing that, you're going to get more ATP because you have a healthier cell. You're going to have healthier mitochondria. I sell in a 2 ounce, a 4 ounce, and an 8 ounce, and in avocado and coconut oil, because this is lipid soluble. So lipid soluble. I have a plethora of different supplements. I have D3, which is very important as a transcriptional cofactor for gene expression. It also helps with apoptosis of cells, and it helps with the absorption of calcium. Vitamin C, which is obviously needed for your skin, but it helps with the immune system. Omega-3, very important for your cardiovascular health. Ashwagandha to control your blood glucose levels. Good Night Formula has tryptophan and melatonin in it. So this is going to allow you to get into REM sleep. When you're in REM sleep more often, your body's going to heal more often. You're going to be more rested, and you're going to be able to solidify your memories. And being in REM sleep will increase the flushing of your cerebral spinal fluid, right? Your central nervous system produces proteins and kind of like waste products, and it gets flushed into the lymphatic system, but it does that during your sleep cycle, more prevalently in your sleep cycle. So if you're sleep deprived, you're actually accumulating more of the waste products that your brain secretes into the cerebral spinal fluid, the CSF. The premium probiotic is a great probiotic that's in a powdered form. So I put this in my hummus every lunchtime, and that's how I take it. Resveratrol, very strong antioxidant, works well synergistically with C60. It also helps to apoptose senesced cells. This is the reason why people that take resveratrol have an anti-aging property. Vitamin B complex. You need your vitamin Bs to prevent neurological dysfunction, macrocytic anemias, and to make sure your electron transport chain is working. Many of the vitamin B vitamins are cofactors for metabolic function. So if you are slightly deprived of your vitamin Bs, you're not going to have the metabolic functioning that takes place, especially in terms of blood cells. Turmeric. This product is a very good product to bring down that inflammation. That's another part of the pillar. So the 3 main pillars of the protocol: reduce reactive oxygen species, reduce inflammation with the turmeric and the ashwagandha, and neutralize pathogens with the structural nanosilver. The the C60 is a great way to reduce the actual reactive oxygen species, so you get that proper cellular health. Zinc and magnesium, you need this as a supplement. I take zinc every day. I double it if I feel sick. Zinc has been found to help to improve the immune system and fight infection and pathogens. Lignans is an important supplement to boost your immune system and your hormonal health. So what I do is I take this with the hummus. I add, because this is in powdered form, I take the lignans and the probiotic together and put it in my hummus. I mix it together. Some people mix smoothies and stuff, and put the powder in that. Collagen, which is also a powdered product, very important to improve your joint health, especially if you had some sort of injury, some sort of nagging joint problem because you used to be a runner or whatever. But it's really, really good also for skin health and improving the dermal layer. When you take collagen with the vitamin C product that I sell, and also apply the nano silver gel on the surface of your skin, what's going to happen when you drink the 64 ounces of water daily that's filtered, no fluoride, you will start to see an improvement of your skin. And what's happening is that you are improving the topical part of the skin and the deeper layers of the skin. So the topical part of the skin is the nano silver gel, okay, and that's going to help heal. Also, you could use the Even Better bar that I sell. But so you have a topical product, Even Better bar or the nano gel, and the vitamin C with the water and the collagen will help to improve the lower layer, the dermal layer, and improve the tightening of the skin and the health, because your skin is produced at a lower level and it pushes up and turns each level as it's getting to the surface becomes more, that there's more keratin in it, and so it becomes more watertight. So it's important to do the topical part, but also the internal part. So the collagen, the vitamin C, and the water helps with the lower part. In addition to helping the lower part of this for skin health, resveratrol will get rid of senescent cells, so it'll improve your basal layer, because you have adult stem cells in the basal layer of your skin. All right, and that's where it's being regenerated. So resveratrol is going to help with that, and the C60 will lower the stress that's been in those cells, especially through UV radiation. UV radiation has damaged your cell, and so it's important to get rid of those damaged cells. Taking the resveratrol helps the senescent, some of these senescent cells, to get rid of them. Also, the D3 is going to help with trying to get rid of these cells that have been somewhat damaged, because that helps with apoptosis. And the C60 is for cells that are somewhat stressed but not fully damaged. It's trying to bring them back by reducing that reactive oxygen species and improving that skin health. So you're working at so many different layers here. You're working at the lower layer of it, and the topical layers of your skin, and trying to target some of the damaged cells from just ultraviolet. So in addition, I also have the digestive enzyme complex. The digestive enzyme complex is great to break down your proteins, your carbohydrates, and your lipids. It's a great supplement to use. It's in a capsule form, and you can take it if you're like someone that is working out and you need to refuel. So this will help to increase your refueling. But as you get older, your pancreas isn't going to be working as well. So supplementing for digestive enzymes is going to help reduce the load on your pancreas. Now, Clarity Factor is a great product to reduce that brain fog, to allow you to think more clearly and have concentration. I take this product twice a day. I take it in the morning and mid-afternoon. And then I also have products that Rainbow Herbals creates here. The Even Better Bar is great for skin health. It's also can be used on cuts and abrasions, minor burns, insect bites, muscle aches. And you'll be amazed at how fast it reduces the pain, especially like a stretched out muscle, or just after a long run, and you apply the bar on aching muscles. You'd be surprised at how fast it actually reduces the pain. But what's even more amazing is that you can apply this on a cut, and you will see that your cut is healing quicker. The product is made from essential oils from the Himalayas. It's a very high quality product, and that's the reason why the price point. In addition, Rainbow Herbals also makes the two types of deodorant, a citrus deodorant and a peppermint lavender. Very high quality deodorants, also made with essential oils from the Himalayas, very high quality. I highly recommend it. Now, I've curated different boxes here, right? And, what people would need to start an anti-aging protocol. If they were only using nano silver, they could do, they could get this box where it would have Max 35, the toothpaste, the silver gel, the probiotic, the silver soap, and the silver lozenges, and you can pick which ones, right? Then I also have skin health for beginners. This is, like I was saying, the vitamin C and the magnesium with the collagen and the gel will improve your skin health. The anti-Fauci box. Now this has all three pillars in it. The neutralizing of the pathogens, which is the Health Max 35, the turmeric, which brings down the inflammation, right? And the ashwagandha will also control the blood glucose levels and bring down that inflammatory response. And then obviously the D3 and the zinc will help with with boosting up your immune system. And the D3, as I said, are cofactors for gene expression and for getting rid of cells that need to go into apoptosis. So I have a lot of different products that I offer on the store. So it's the-studio-reykjavik.com. So please, if you can, donate on Stripe or PayPal on the homepage. Or you can donate on Buy Me a Coffee. The link is in the description of this video and all my videos. Please subscribe to all my channels. I have 6 different channels. I have 3 YouTube channels. Please subscribe to all 3 YouTube channels. Make sure you click the notification button. I am heavily censored on YouTube. I can't tell you how much censored I am, because every single time I leave a video up that is somewhat radioactive on the crisis, it will get deleted and I get a strike. All right. So I move all of my content also over to Brighteon, Bitchute, and Rumble. Please go there and subscribe so you can see the content that is censored on YouTube, and also see my new content. I will premiere on YouTube, and if I can keep it up on YouTube, I will. But you have to subscribe to all 6 channels to be able to see my content. That is the only way I can get around the censorship. Now, in addition, please go to my Patreon channel and subscribe to help support the news coverage. For a nominal fee, you can subscribe to Patreon, and you'll be able also to see the content there. So these are the different mechanisms that you can help to support my news coverage. The most important is for you to go to the store and get the health products and follow the protocol that I have mentioned: neutralize pathogens, reduce reactive oxygen species, and lower down that inflammatory response. And then you add to that, like the probiotics and the multivitamins and the lignans and the collagen and all this other stuff. But if you follow my lead, if you follow what I'm saying about proper supplementation, proper diet, proper exercise, not too much exercise, but nice moderate amount of exercise, right? Proper mental health, get enough sleep, make sure you don't get too depressed, make sure you challenge your mind by reading, and have those more intelligent conversations with people, and don't be in a rage, because that's not good for your health. So if you have this, proper diet, proper exercise, proper sleep, proper challenging of the mind, proper nutrition, you are going to improve your immune system and not be a victim of the system. Because the system, meaning the poor food that's out there, the fast food, the GMO food, the fluorinated water, the negativity in the media, these things are all chipping away at your immune system. And if you do things in your life on a regular basis that improves your immune system, then you're going to fare better. So thank you for supporting me. Make sure you subscribe to all my channels, and I will do the 4th video dealing with pseudouridine and how it's correlated or used in cancer cells to stabilize the protein synthesis. Thank you for listening, and have a nice day.